When you look at a plant, you probably see something, well, kind of passive, right? Just sitting there, stuck in one spot. But what if I told you that beneath that green, calm exterior, there's a high stakes genetic gamble going on? It turns out when things get tough, like a heat wave or a drought, plants don't just endure it. They actively start rewriting their own DNA. So let's dive into this absolutely wild world of rapid plant evolution.
So here's our game plan. First, we're going to tackle the big adaptation puzzle that every plant faces. Then, we'll unpack a really important genetic tool called copy number variation. After that, we'll look at a fascinating stress test experiment that really puts this idea to the test. We'll even meet these little genetic movers and shakers called transposable elements, see how genomes actually get reshuffled, and then we'll bring it all back to the real world and the food on our tables.
All right, let's kick things off with the first piece of our story. This central problem, this dilemma that every single plant has to solve.
I mean, really think about this for a second. A heat wave hits, the soil gets too salty, a new disease pops up. A plant can't just pick up its roots and leave. And yet, flowering plants, angiosperms, have somehow managed to spread across basically every corner of the planet. That tells us they must have an incredibly flexible genomic toolkit that lets them adapt, and way faster than we used to think. So the real question is, what's in that toolkit?
Well, to find the answer, we need to look at a powerful and honestly an often overlooked type of genetic change.
It's called copy number variation, or CNV for short. Now, you probably heard about mutations as these tiny little typos in the DNA code, right? Like one letter getting swapped out. But CNV, whoa, it's on a whole other level. It's not about changing a letter, it's like grabbing an entire paragraph from the genetic instruction book and either ripping it out completely or just hitting copy-paste a few extra times. We're talking about major structural changes, not tiny edits.
And this isn't some rare biological fluke either. CNVs are a fundamental driver of evolution. They create a ton of diversity within a species, and that provides the raw material for natural selection to work with, especially when the environment goes haywire. They've been linked to everything from diseases in humans to the very traits that make our crops grow. So the big takeaway here is, if you want to understand rapid adaptation, you have to understand CNV.
Okay, so to really see this in action, scientists set up this brilliant experiment. It's basically like watching evolution on fast forward. And they use the classic lab workhorse for this, the little model plant, Arabidopsis thaliana. So here's how they did it. They started with plants from a single common ancestor, making sure they were all genetically identical at the beginning. Then they split them up. You had a control group, chilling at a comfy 22 degrees Celsius. And then you had the stress groups, one at a chilly 16 degrees, another in the hot seat at 28 degrees, and a third group got sprayed with salicylic acid, which kind of tricks the plant into thinking it's being attacked by a pathogen. For 5 whole generations, they'd pick the most successful plant from each group, the one that made the most seeds, and use it to parent the next generation.
Okay, so 5 generations later, how in the world do you see what's actually changed in the DNA? Well, they used this really clever technique called comparative genomic hybridization. It's a fantastic way to visually compare 2 genomes. You basically extract DNA from your stress plant and your normal reference plant. Then you tag each one with a different fluorescent dye, say green for the test plant and red for the reference. You mix them all together and pour them over this special chip, and the colors tell the story. If a spot on the chip glows yellow, it means both colors are there equally, no change. But if you see a bright green spot, that's a duplication. The stressed plant has more copies of that gene. And if a spot glows red, that's a deletion. It's gone. Super visual, right? And when they looked at the results, a really fascinating pattern emerged. Check this out. The plants in the cold and the ones faking a disease both showed a net loss of genes. They were mostly deleting stuff. But the plants cooking under high heat stress, they were different. They had deletions too, sure, but they had way more gene duplications than the other groups. This points to a totally different adaptive strategy kicking in.
This was a huge finding. The 28-degree lineage, the one under the most severe stress, also had the most unstable genome. In fact, siblings within that group were more genetically different from each other than in any other group. This draws a direct line between environmental pressure and the rate of genetic change. It's like stress isn't just a filter for random changes, it's hitting the gas pedal on change itself. So what's actually causing all this crazy reshuffling? Well, a big part of the answer involves these little rogue agents living right inside the genome. Let me introduce you to transposable elements, or TEs. You might know them by their cooler name, jumping genes. And that's exactly what they do. These are pieces of DNA that can literally cut themselves out of one spot in the genome and paste themselves into a completely different one. And in doing so, they can duplicate genes, delete them, or just mess them up. They are powerful, powerful engines of genetic variation.
So, sure enough, when the researchers looked closer at the genes involved in these CNVs, transposable elements were everywhere. In the cold weather plants, they made up a massive 40% of all the genes in those changed segments. But here's where it gets weird. In the most stressed out 28-degree group, TEs only made up about 10% of the genes in those stable changes. What does that suggest? Well, maybe as the stress gets really intense, the evolutionary response becomes less random and starts to zero in on more specific, functional genes.
So let's get into the nitty-gritty. How does this reshuffling actually happen at a molecular level? We're going to zoom all the way in to see the specific cellular mistakes that cause these huge genetic shifts.
So, generally speaking, there are two main culprits behind CNVs. First, you've got errors that happen during recombination. That's the process in sexual reproduction where chromosomes line up and swap bits of genetic material. Second, you have errors during replication, which is just the basic act of copying DNA every time a cell divides. Both of these are totally fundamental processes, but one tiny little slip-up in either one could have massive consequences.
Let's start with a recombination error, specifically something called unequal crossing over. Normally, when chromosomes are getting ready to swap genetic info, they line up perfectly, gene for gene. But sometimes, two regions that just look similar but aren't actually in the same spot get paired up by mistake. So when the swap happens, it's lopsided. One chromosome ends up with a duplicated piece of DNA, and the other one loses that exact same piece. It's a perfect illustration of how a simple mistake can create both a duplication and a deletion all at once.
Okay, now for a replication error. This happens when the DNA copying machine itself messes up. Imagine the machinery is unzipping and copying a strand of DNA, and then it just hits a snag and stops. It stalls. In that little moment of confusion, the new DNA strand that's being made can actually detach and then mistakenly reattach to a nearby sequence that looks kind of similar. When it starts copying again from that new spot, it can end up copying a whole section of DNA that it already copied. It is literally a real-time copy-paste error at the molecular level. Now, this is not just some weird thing that happens in a lab. The fingerprints of copy number variation are all over the plant world, and they're especially important in the crops that we all depend on to eat. Just look at these examples. In wheat, changing the number of copies of certain genes is what allowed it to adapt to different day lengths and cold winters as humans carried it across the globe. For maize, having extra copies of a gene called MATE1 lets it grow in acidic soils that are full of toxic aluminum. In barley, more copies of the BOT1 gene helps it deal with boron toxicity. And in soybeans, having multiple copies of a set of genes called the RHG1 locus makes it resistant to these really destructive pests called nematodes. I mean, these aren't small changes. These are the critical adaptations that allow our food to grow where it grows. So what's the big picture here? Is the plant somehow intelligently making these changes? Not quite. And this quote sums it up perfectly. It's a high-risk strategy. The cell has no idea if duplicating a gene is going to be good or bad. But when a particular gene is being used a lot because of some stress, that area of the DNA just becomes a hot spot for these kinds of errors. The plant is essentially just rolling the dice more often on the very genes that are most important for its immediate survival.
Which brings us to this one last really fascinating question to think about. We usually see stress as a bad thing, right? A bug in the system that causes things to break down. But everything we've looked at today suggests another possibility. What if, in the grand story of evolution, environmental stress is actually a feature? What if it's a built-in mechanism that doesn't just passively filter out the weak, but actively hits the accelerator, boosting genetic innovation and speeding up a species' ability to adapt and survive in a world that is always changing?